Inside a living cell, the environment is anything but empty. Proteins, ribosomes, RNA, and organelles are packed together in a dense, chaotic soup. If you were to shrink down to the nanoscopic scale, you would find a world more crowded than a subway at rush hour. And yet, despite this extreme molecular congestion, biological processes like enzyme reactions, signal transduction, and gene expression occur rapidly and efficiently. How does nature maintain order in such a seemingly chaotic environment? One would expect that a highly crowded cellular environment would slow down molecular diffusion, making it difficult for proteins and signaling molecules to move around and interact. After all, in classical physics, the more obstacles in a system, the more resistance to motion. Yet, paradoxically, cells do not experience a breakdown in function due to overcrowding. In fact, many biochemical reactions accelerate under these conditions. How is this possible? The key lies in the non-equilibrium properties of molecular motion in the cell. Unlike the simple random Brownian motion seen in dilute solutions, molecules inside a cell move via anomalous diffusion, where motion is influenced by energy-driven transport mechanisms and spatial organization. Rather than relying solely on passive diffusion, cells use molecular highways, such as cytoskeletal networks, motor proteins, and vesicle transport systems to actively direct molecules to their targets. Imagine a crowded city where, instead of everyone walking randomly, there were high-speed trains and conveyor belts moving people exactly where they need to go. Another surprising aspect of molecular crowding is that it stabilizes biomolecules. Proteins often require precise folding to function correctly, but in dilute solutions, they can unfold or aggregate incorrectly. The crowded cellular environment helps maintain proper protein folding by limiting excessive movement and favoring compact, functional conformations. In this sense, molecular crowding acts as a molecular chaperone, assisting proteins in maintaining their structure under conditions where they might otherwise degrade. Yet, the paradox deepens. If crowding is so beneficial, why don't cells just become even more packed to increase efficiency? The answer lies in the delicate balance between order and flexibility. If a cell were too densely packed, movement would become too restricted, leading to metabolic gridlock. Cells solve this by maintaining compartmentalization, where biomolecules are organized into distinct regions, ensuring that local concentrations of reactants are high without causing complete congestion. One fascinating analogy comes from human-designed systems. Think of a well-organized factory. If raw materials were randomly scattered, production would be inefficient. But if everything were crammed into one spot, workers wouldn't be able to move. Instead, the most efficient factories have workstations with specialized tools and assembly lines directing flow, just like how cells use organelles, phase separation, and transport pathways. So what does this tell us about life at the molecular level? The molecular crowding paradox highlights how biological systems use constraints to their advantage. What might seem like an obstacle, extreme molecular density, actually enhances efficiency through self-organization, compartmentalization, and active transport. This insight doesn't just reshape how we understand biochemistry, but also has major implications for nanotechnology, drug design, and synthetic biology. Could we learn from cells to design artificial crowded environments that enhance chemical reactions for industrial or medical applications? Could understanding crowding effects help us treat diseases like Alzheimer's, where protein misfolding plays a key role? And most importantly, what else does nature accomplish that seems paradoxical at first, but turns out to be a brilliant adaptation for survival? Cells are not dilute solutions, but highly packed environments where macromolecules occupy up to 40% of the available volume. This molecular crowding increases local concentrations, accelerating reaction rates that would otherwise be diffusion limited. Despite this congestion, biomolecules exhibit remarkable efficiency in finding interaction partners, forming organized structures without chaotic interference. Enzymes in metabolic pathways, such as glycolysis, function with extreme efficiency despite the high density of surrounding proteins and organelles. This is similar to a busy city street where, despite heavy traffic, pedestrians and vehicles still move efficiently due to self-organizing patterns. While diffusion is slower in crowded environments, cells compensate by employing active transport mechanisms such as motor proteins and cytoskeletal highways to ensure targeted molecular delivery. Kinesin and dynein motor proteins transport vesicles and organelles along microtubules, ensuring timely arrival at their destination. This is like a factory conveyor belt system that speeds up the delivery of parts to specific assembly stations instead of relying on workers randomly searching for materials. Molecular crowding alters macromolecular behavior by inducing entropic forces that drive self-assembly and organization. The excluded volume effect forces proteins, nucleic acids, and other molecules into specific spatial arrangements that optimize function. This effect enhances the efficiency of ribosome formation and large protein complexes. This is like packing luggage into a suitcase. Items naturally arrange themselves into the most spaceless configuration without deliberate or crowded environments, influence protein folding dynamics by stabilizing native conformations and reducing misfolding. The excluded volume effect favors compact functional protein structures, preventing aggregation and promoting stability. Heat shock proteins, HSPs, assist in protein folding under stress conditions where crowding effects are amplified. This is like a tightly packed bookshelf where books must be arranged neatly to fit. Random placement leads to inefficiency and disorganization. Molecular crowding significantly alters reaction kinetics by increasing the effective concentration of reactants and reducing the available volume for diffusion. Enzyme-catalyzed reactions become more efficient due to higher collision probabilities, but excessive crowding can hinder diffusion-limited processes. Cells regulate local crowding dynamically to balance reaction efficiency with proper molecular distribution, as seen in microdomains where metabolic enzymes cluster to enhance reaction rates. This is similar to a small cafe with limited seating. While customers interact more frequently due to proximity, excessive congestion can slow service. The cell nucleus, a highly crowded environment, must efficiently manage RNA transcription, splicing, and export, despite the dense packing of chromatin and proteins. Molecular crowding enhances the efficiency of RNA processing machinery by keeping essential factors in close proximity. Nucleoli, where ribosomal RNA is synthesized and processed, act as phase-segmented condensates that maintain high concentrations of necessary enzymes. This is like a shared workspace where employees sit in close proximity, ensuring rapid communication and teamwork efficiency. Cells actively modulate their internal crowding by adjusting cytoskeletal structures, osmotic balance, and phase separation mechanisms. Under stress conditions, they reorganize the cytoplasm to prevent excessive molecular congestion and optimize function. For example, cells exposed to hyperosmotic stress shrink temporarily, but restore their volume by regulating ion channels and osmolytes to maintain homeostasis. This is like adjusting furniture in a crowded room. Rearranging elements can create better flow and improve movement efficiency. Phase separation allows cells to form membraneless organelles, which compartmentalize biochemical reactions in crowded environments. These biomolecular condensants concentrate macromolecules without physical barriers, enabling rapid molecular interactions and efficient regulation of gene expression, stress responses, and signal transduction. P-bodies and stress. This is like oil droplets in water spontaneously forming separate domains, allowing different reactions to occur in distinct environments without rigid walls. Understanding molecular crowding is crucial for designing synthetic cells and nanotechnology-based drug delivery systems. Artificially engineered biological systems must account for crowding effects to ensure proper molecular interactions and diffusion rates. For instance, synthetic protocells designed for biochemical reactions incorporate molecular crowding principles to optimize enzymatic efficiency. This is similar to designing a high-efficiency warehouse where storage density is maximized while maintaining easy access to critical components.